Good morning, everyone. So I'm going to talk about some of the research projects I did for my PhD at the University of Sussex, and this is in the southeast of England. So my PhD was all about free-living honeybee colonies, and by definition, these are colonies that are unmanaged, living in a nest site of their own choosing, and also referred to as wild or wild living, unmanaged, or even feral. And the only difference there is semantics, really, but free living is the most politically correct term for them. So free living colonies have been traditionally viewed as swarms that have just escaped from nearby apiaries, so essentially a byproduct of beekeeping. And as a result, they received relatively little attention in the scientific literature. And we know very little about their basic ecology, so things like how many there are and whether they're actually surviving from year to year. So my research has sort of attempted to address some of these gaps in the knowledge through both original fieldwork and also syntheses of existing data. So my talk's going to be divided into two main areas. The first covers the basic ecology of free-living colonies, so the occurrence, density and survival. And I first look at this on a global scale through a review I did a few years ago, and then move on to my field research in southeast England. The second part of the talk is going to be looking at potential limiting factors. And so during my PhD, I developed a couple of methods that basically help quantify the quality and quantity of available nest sites to help evaluate their role in limiting free-living colonies. I thought I'd start by just asking, why should we study free-living colonies? So a typical honeybee population will consist of both free-living and managed colonies, as Norman said, and they both exchange genetic material through mating over quite long distances, and they also exchange swarms. So the managed colonies are often exposed to artificial selection to some extent for traits like tameness or honey production, whereas the free-living colonies are exposed to pure natural selection. So by studying the free-living colonies, specifically their density, or the number of colonies per square kilometre, we can get a sense of how important natural selection is in driving the health of the whole honeybee population in that area. And you might expect adaptations to the local environment and novel pests to be more prevalent in areas with high densities of free-living colonies. So I looked at this on a global scale by reviewing all of the published literature that have quantified the density of free-living colonies. I found a total of 41 locations worldwide that have quantified density either through direct surveys, which are the locations in red, and these are studies that have just gone and looked for colonies in defined areas. And there are also a few studies that used an indirect method that involves using population genetics, so sampling drones at congregation areas and genotyping them to see how many different colonies they've come from. And the countries in green here are all those with data on managed colony density. So I should briefly mention that the honeybee, the western honeybee, Apis mellifera, is native to Europe, Africa and the Middle East. As you can see, it's been introduced to all other habitable continents. And free-living colonies also occur pretty much everywhere where honeybees are managed, with the exception of Asia, potentially. There's a conspicuous absence of free-living colonies of the western honeybee, and for various potential reasons. So I found that density really did vary quite significantly, and on this graph here I showed density divided into the 5 main geographical regions, and you'll notice that the density is on a log scale here, so it occurs over several orders of magnitude, all the way from around 0.1 colony per square kilometre in some parts of Europe, Central Europe, all the way up to 20 colonies per square kilometre in parts of sub-Saharan Africa. But the overall global average is around 2.5 colonies per square kilometre. And I found that the densities reported in Europe were significantly lower than all other geographical regions. And this was even after controlling for factors like climate. So it's not just because it's colder in Europe. And there are several potential reasons for this. And they include things like habitat loss and a more intensive apiculture in Europe. So a lot of the densities reported in Europe come from German researchers Patrick Cole and Benjamin Rutzmann. And in their study system, they were looking at free-living colonies that occupied old woodpecker nests that had been excavated in beech trees in these large forests in Germany. And they're excavated by the black woodpecker, which is the largest woodpecker in Europe, and they're not actually present in Britain. But they excavate large enough cavities to contain a honeybee colony. And these researchers monitored 100 colonies or so for around 4 years and recorded a really low density to start with, but also a really low winter survival rate of around 16% on average. But when you're looking at just the summer densities, I mean, they're very low, as I say, around 0.2 colonies per square kilometre. But if you're just looking at the summer densities, they look relatively stable. And that's because the cavities are getting recolonised by swarms from the surrounding areas, be it from other wild colonies, free-living colonies, or managed colonies. So although the population looks stable, in reality it is dependent on the annual immigration from swarms from the surrounding area. And again, there's several potential reasons for these low densities and survival recorded in German beech forests, several of which they investigated in subsequent studies. And they're actually able to rule out one of the more obvious factors, which is disease. They showed that disease levels didn't differ at all between colonies that survived and those that died. And they found that the colonies really weren't living long enough for disease to become a problem. So another factor is the quality of the nest sites they were occupying. As I mentioned, they're excavated by a large woodpecker, which creates a really large entrance hole around 10 centimetres on average. And this makes the cavity really hard to defend from predators like other woodpeckers over the winter. And they covered some of the cavities with mesh and found that those colonies survived at significantly higher rates, although it was only a marginal increase. So another factor they investigated was food supply. The beech forests that these colonies are living in produce very little nectar, and that's because beech trees are wind pollinated. And they actually found that colonies living on the edge of the forest had a significantly higher survival, and these were the colonies where the bees were able to forage in surrounding cropland. So habitat loss is definitely a factor, contributing to these results. But it doesn't explain why similar survival rates and densities have also been recorded in urban areas in Germany, where the bees are foraging and nesting in different habitats. So overall, it seems that only a small percentage of honeybee colonies in Europe are living in the wild and exposed to natural selection, and that beekeeper management may play a more important role in determining the health of the whole honeybee population. However, this was a very large-scale analysis, and it's highly likely that there are hotspots in parts of Europe where free-living colonies are more numerous and have a better chance of surviving. And I thought that a good place to investigate this was the English countryside, Because although we don't have a lot of large forests, we have a really large collection of what we call veteran trees, which are trees that have survived previous changes in land use. And have been recognized for either their size, age, or value to wildlife. And one of the important things they provide for wildlife is cavities where animals can live. The veteran trees are quite extensively mapped across the UK, and this is largely thanks to a citizen science project run by the Woodland Trust called the Ancient Tree Inventory, or ATI. And the ATI has data on over 200,000 veteran trees across the UK. This includes things like the species of the tree, the circumference at breast height, so the trunk size, and also the veteran status, so ancient, veteran, or notable. Of the ancient trees, which are shown in orange on this map here, being the oldest in general. And all of this data is available to the general public through their interactive map. I've provided a link here in the bottom right. So you can, look at where all the old trees are, where you're living, and go and search them. And that's exactly what I did a few years ago. I used the ATI to search over 1,000 veteran trees across 16 sites in southeast England. These are in the counties of Sussex, Kent, Surrey, and Greater London. And this was basically to determine how many of these trees, or what proportion, were occupied by free-living colonies. And I mainly searched trees in the spring, which is quite a good time to look for free-living colonies, because the trees aren't in leaf, so their nest sites aren't obscured by foliage. And it also means that you're only finding established colonies that have survived the winter. And these are more ecologically interesting. So overall, I found that out of the 1,000 trees I searched, only around 21 or 2% were occupied by free-living colonies, which might seem like quite a low occupancy rate, but I'll come on to that later and why it's actually quite significant. And after the study was completed, I continued to monitor the colonies I'd found for several years at a selection of the sites. So I found that occupancy had quite a strong correlation with the size of the tree, so the diameter at breast height, which is a common metric used in arboriculture. So this was quite a surprising result, because the trees on the database already had a really large mean diameter, average diameter of around 1.3 meters at breast height. Well, I found that trees containing bees had an even larger average diameter of 1.7 meters. Well, I should say there's quite a lot of variation around this average. So on the lower end, I found a colony nesting in a very slender Scots pine, which had a diameter of around 0.5 meter at breast height. And then on the same site, this is Petworth Park in West Sussex, I found a colony nesting in this 900-year-old oak tree with a diameter of around 3 meters at breast height. I found that most of the colonies were nesting in oak trees, but this was expected because they're the most common tree on the database. I also found quite a few colonies nesting in sweet chestnut trees, or castanea. And this was more than expected just based on random sampling. And it's probably due to the fact that the sweet chestnut trees had the largest mean diameter of all tree genera on the database. And they also had quite a high chance of having what looked like suitable cavities. So the ATI is a really useful resource for anyone interested in honeybees or other cavity nesting species, but it's far from complete. And there was a recent study that estimated there are 2 million veteran trees in England alone, which is 10 times more than are currently listed for the whole of the UK. But based on this estimate and our occupancy rate of 2%, we estimated that there could be upwards of 40,000 free-living colonies nesting in veteran trees in England alone. And this doesn't even include colonies nesting in other structures like buildings and rock faces. And for comparison, the MBU estimates there are around 250,000 managed honeybee colonies in the UK. So it's highly likely that free-living colonies are a significant component of total colony numbers. This is especially the case in areas with high densities of veteran trees. So now I'm going to move on to the long-term monitoring that I've been doing on some of the sites I mentioned. And all these sites are examples of what I call landed estates. And this is a term I use to describe the large area of semi-natural habitat surrounding a stately home. And it includes habitats like wood pasture, parkland, and deer parks. So there are thousands of these estates across the whole of the UK. They only make up 2% of land cover in England. Many of these estates have been owned by the same families for centuries, and as a result, they've undergone very little change in land use, and veteran trees have been able to persist at quite high densities. They're also often open to the public, which makes them really great places to look for free living colonies, and I always recommend that people, check out National Trust properties if they're interested in finding free living colonies. There are many ways to find free living colonies, but by far the most entertaining is a technique called beelining, which basically involves following forager bees back to their hive from an artificial food source. And to do this, you'd catch bees foraging on a flower patch or something, and then train them to a sugar feeder. And you'd mark bees with a bit of paint so you can recognize individuals, and then record the direction from which they depart from your artificial feeder and the amount of time it takes them to come back. And you can use these two values to get a general idea of where the nest site is. And once you know that, you can move the feeder in the direction of the nest site until you're getting return times of a couple of minutes or so. And at that point, you're within 100 meters of the colony, and you can basically search that smaller area. So I've attempted beelining on various occasions. And on this occasion, I did a variation of beelining where you move laterally to where you think the nest location is, so that you can sort of triangulate the location without moving into like a dense forest. And I would say that beelining is a lot of fun, that it's extremely time consuming, and depending on where you're doing it, you have just as much chance as ending up at an apiary than a free living colony, which is unfortunately what happened on this occasion. But so also you end up having to search anyway. So I found that the most effective way of finding colonies on these estates is by cutting out the middleman and just going straight to the potential nest sites. And on these estates, the total tree densities are really quite low. So it is actually possible to search every tree over a threshold diameter. And I use, half a meter at breast height as the, minimum tree that I look at, because that's the smallest tree I've ever found to have bees in it. But I also use another method, which is basically just asking local rangers and land managers whether they know of any colonies. But the majority of nest sites that I've found in southeast England are just by searching veteran trees, some of which are listed on this database I mentioned. So here's an overview of the nest sites I was monitoring for my PhD. And there's a total of 63 at 6 sites across southeast England. And I did find, more than this, but this was just the nest sites that I was monitoring. And the largest area I surveyed was 2.6 square kilometers, at a estate called Petworth Park in West Sussex. They found a total of 23 nest sites at this estate, all of which were nesting in trees, which is the honeybee's natural nesting habitat. I should say that 90% of the nest sites I was monitoring were in trees. And this map on the right here shows all of the estates in England with at least one tree listed on the ancient tree inventory. And the 6 I surveyed are shown in red there. And I've also given buffers of 2, 5 and 10 kilometres around each estate, just to give you an idea of how widespread they are, even in the southwest. So I've been able to collect quite a lot of data for each of these nest sites. And this has largely been thanks to a bit of equipment, a large telescopic pole which extends all the way up to 12 meters. And it's quite rigid, so you're able to put various bits of equipment on the end of it to get it up to the nest site. And most recently I've been using a 3D scanner to get really detailed measurements of the cavity entrance. And you can also just use it simply to measure the height of the cavity. And the nest sites I was monitoring ranged all the way from ground level, so 0 meters above the ground, all the way up to 17 meters with a median of around 7 meters. And I found that the entrance size was really quite variable. It had an average of around 6.5 centimeters in diameter, which is relatively large, but not as large as the black woodpecker cavities they were monitoring in Germany. I also found that entrances were orientated in random directions, which was quite surprising because you might expect them to be, biased towards the southeast, where they'd get more sun exposure. But in terms of the monitoring, I visited nest sites 3 times a year, and this is sort of the minimum requirements to accurately determine survivorship. So the first survey was in spring, before the swarming season, and this was to quantify the number of colonies that survived over winter. The second survey is done in the summer, after the swarming season, and this was to determine the recolonization of inactive nest sites by swarms. And the third survey was in autumn to quantify mortality over the summer and the number of colonies going into winter. So in each survey, I would check for directional flight, pollen import, and defensive behaviour, which were all signs that a nest site is occupied by an active colony. And using the telescopic pole, it was sometimes possible to actually see inside the cavity and determine survivorship with 100% certainty. And these photos here are of a nest site taken at 4-month intervals. And they show a colony at its maximum population size in July, and the population size significantly decreased in November until in March it died and had been sort of robbed out by birds and wax moths. And then the nest site was then recolonized by a swarm the following summer. And this isn't to say that this is the typical life cycle of a free living colony. It just gives an example of what can happen over the year. And I've found nest sites which have been continuously occupied by a colony for several years. So here's an overview of the monitoring that I'd done over a period of 3.5 years, so from autumn 2021 up until spring 2025. And the graph here shows the changes in colony density during the study period. And the spring surveys are in green, summer's in blue, and the autumn is in red. And I found that most of the colonies would survive summer, which is what you'd expect, around 90% summer survival, but in a much lower proportion survived winter, at around 50% on average. And by multiplying these two survival rates together, you get an annual survival rate of 41%. Which might not seem like a lot, especially from a beekeeper's perspective, where an annual survival rate of 40% just wouldn't be viable if you wanted a good honey crop. But the thing with free-living colonies is that nobody's stopping them from swarming. And I calculated that the colonies that survived until spring would only need to produce an average of 1.4 swarms each every year to basically offset this high mortality over winter. So it's highly likely that this population is actually viable. And studies have shown that when left unmanaged, a honeybee colony will typically produce 2 swarms every year, so a prime swarm and an after swarm. And I recorded an average density over the study period of 2.5 colonies per square kilometre, which you might remember is actually the global average. But more importantly, it's 10 times the density that has been recorded elsewhere in Europe. And quite interestingly, it was very similar to the density of managed colonies in the local areas surrounding these estates. So this has quite important implications for beekeeping in these areas. So I've shown that these colonies are not just swarms that have escaped from apiaries, and they may even represent additional genetic variation that can be used to improve beekeeping. This genetic variation is going to be accessible to beekeepers just by catching swarms or using locally mated queens. We'll talk more about the project we've got lined up in the southwest in the second session. So now I'm going to talk about the second part of my research, which is all about limiting factors, specifically nest sites as a potential limiting factor. So nest sites are often assumed to limit free-living colonies, as with a lot of cavity nesting species. And this is understandable because in the case of the honeybee, they have really quite specific nesting requirements. They need a cavity of at least 15 litres to house the quantity of honey needed to survive winter in northern latitudes. And on top of this, they need a cavity with quite a small entrance so that they can effectively defend the honey stores and keep the cavity warm over winter. So these types of cavities take centuries to develop naturally through processes like decay in old trees. And as a result, they occur at much lower densities in Europe than other geographical regions due to the longer history of deforestation. But that being said, honeybees are not obligate forest dwellers, and they will readily use cavities in other structures like buildings. And I've also shown that they nest at naturally low densities in temperate regions like Europe, densities of around 1 or 2 colonies per square kilometre, which suggests that the demand for cavities is actually quite low. The role of nest sites in limiting free-living colonies also depends on the cavity supply. So how many are there in the environment? And the truth is we really don't know much about that. And the reason we don't know much about that is because it's really hard to quantify, especially in urban areas where there's just a lack of access and quite a large number of potential places that honeybees could nest. So as part of my PhD, I developed an indirect method which uses honeybee swarms to assess nest site availability. So as many of you will know, the honeybees use a behaviour called the waggle dance to communicate the location of profitable resources to their nestmates. And in the context of swarming, scout bees will produce waggle dances on the surface of the swarm cluster. And researchers have been able to record these dances and then decode them to provide insight into the process of nest site selection. So Tom Seeley wrote a brilliant book about this called Honeybee Democracy. But what we wanted to investigate was whether swarm dance decoding could be used to provide ecological information about the availability of nest sites in an environment. So a waggle dance consists of 2 phases. We've got the waggle run and the return phase. And it's the waggle run that we're interested in because it contains the spatial information. So the direction and distance to a nest site is encoded in the angle and duration of the waggle. But it's quite a noisy signal, so there's a lot of variation within a single dance. And we were able to simulate this variation to produce heat maps showing where the advertised location is most likely to be. And that's shown as these green and yellow pixels here. And the red cross is where the swarm was set up. This is on the University of Sussex campus. And by averaging those pixels, we can get a point location of where the nest site is most likely to be. And I refer to these as nest locations and not nest sites because there is this degree of uncertainty. So it set up a total of 14 artificial swarms at 2 study areas in East Sussex. And they mainly consisted of farmland with some urban areas and also patches of woodland. So they sort of represent, a typical mixed rural and urban landscape in southern England. The swarms advertise a total of 56 unique nest locations, and they occurred at distances of up to 11 kilometres from where the swarms were actually set up. But most of them were within 1 or 2 kilometres, which is also the typical foraging range of honeybees. So each of the points here is a nest location, and the colour corresponds with the swarm that advertised it. And locations with 2 colours were advertised by multiple swarms. And we found that 32% of the nest locations were advertised by multiple swarms, so there is actually quite a lot of overlap. We were able to use this overlap to estimate the total number of nest sites in the study area. So this includes the locations that weren't advertised. And we did this using a form of mark recapture, which is a common method used in ecology. We'd multiply the locations advertised by the first swarm by those advertised by the second swarm, and then divide this by both. And the logic behind this is that if potential nest sites are abundant, then 2 swarms are less likely to advertise the same set of nest sites. So this might sound a bit complicated, but we did this with 6 pairs of swarms, and overall it generated a figure of 3 suitable cavities per square kilometre. This is likely quite a conservative estimate of nest site availability for various reasons, but the main one being that we set up these artificial swarms after the natural swarming season, and this was to avoid interference with natural swarms. But it also meant that our swarms were looking for nest sites during the period of highest prior occupancy of cavities in the study area. So this figure of 3 suitable cavities per square kilometre is in addition to those already occupied. And even this conservative value would be enough to support a typical honeybee population density of 1 or 2 colonies per square kilometre. So it suggests that nest sites may not be limiting in these sort of mixed urban-rural environments. That being said, a greater excess of suitable cavities may be needed to offset things like cavity mortality, which is basically a term I use to describe the number of cavities that are lost every year for various reasons, such as a tree coming down or the entrance hole completely closing. And I worked out that 6% of nest sites are lost every year for these reasons when I was doing my monitoring. So more cavities may also be needed to sort of compensate for occupancy by other cavity nesting species, of which there are around 40 in the UK, most of which are birds. And I should note that they have quite different nesting requirements to honeybees, so there's unlikely to be a direct competition between honeybees and other cavity nesting species, but it probably does occur to some extent. And lastly, the most important factor here is that more cavities may be needed to compensate for variation in cavity quality. The limitation of this indirect method using artificial swarms was that we weren't actually able to find the nest sites that were being advertised and assess quality firsthand. So we know that these cavities were high enough quality to be advertised by scouts on a swarm, but we don't know if they were high enough quality to actually permit winter survival. So this is where my most recent research and the last thing I'm going to talk about today comes in. There's a more direct method of assessing the quality of cavities in trees using a 3D scanner. And when I talk about quality, I'm usually talking about cavity volume, because this is really the most important cavity parameter that bees will assess when determining the quality of a cavity. And Tom Seeley has shown that swarms show really strong preferences for cavity volume. When given a choice, they'll choose a cavity of 40 litres over 10 litres or 100 litres. And for reference, a commercial brood box, which is shown here, full of wild comb, this is around 45 litres. So it sort of represents an ideal nest site in the northern latitudes with a long winter. So cavity volume may be important, but again, it's really hard to quantify. It usually requires direct access to the cavity, which involves climbing trees, which is dangerous. But on a field trip to Spain, we were able to access the cavities with minimal climbing because they were all quite low down in these cork oak trees. And we estimated volume just by inserting pieces of wire into the cavity and then using the volume equation for a cylinder or a ellipsoid. This only gives a very crude measurement of volume. So I developed a method that uses a pole-mounted 3D scanner, which allows cavities, the volume to be quantified from the ground level, so you're not having to climb the tree. I use a scanner that's mainly used in for quality control on assembly lines and stuff like that. So it has quite a short scanning range of 7 to 50 centimeters, but within this range it can measure objects with sub-millimeter accuracy. So this means that it actually has applications beyond just measuring cavity volume. And this is an example where I used it on an active honeybee nest site. And you can see from the depth map here that you can actually make out individual bees and even the size of the combs. So, it's potentially quite a interesting technique. I needed to get a way of getting the scanner inside the cavity. So I designed this motorized scanner mount, which consists of 4 3D printed components, which are put on the end of the telescopic pole I mentioned earlier. It basically enables the scanner and a light source to be inserted inside the cavity and then rotated 360 degrees to build up a complete image, 3D image of the inside of a tree cavity. And this is an example of where I've used it on an inactive nest site, which is where it's mainly meant to be used. So this cavity was only occupied for a couple of months, and some of the combs are still intact there, and you can actually make them out on the 3D scan. So this image here is a digital 3D model of the cavity, and it consists of around 10 individual scans that were taken at different points during the 360-degree rotation. And using special software, you can generate a watertight 3D model that can then be measured. And interestingly, this cavity was really quite small, at only 13 litres. So this is, half the size of a nuke box or something like that, and it probably explains why the bees didn't last very long inside. So I won't go into too much detail here. I'll just say that I tested the method on 6 physical cavity models that ranged in size from around 8 litres here all the way up to 22 litres. And this represents the transition from unacceptable to acceptable by honeybee swarms in northern latitudes. And basically I showed that this method can estimate error within this, or estimate volume within this range with less than 10% error. So it's, produces half as much error as that wire method I mentioned earlier. So not only is it more practical, you can do it from the ground, it's also more accurate. But there are a few limitations, the main one being that the scanner won't fit in entrances smaller than 5 centimeters. It's also mainly restricted to vacant cavities. The second point isn't really much of an issue because it's meant to be used on vacant cavities to see whether they're actually suitable to swarms. But yeah, it leads on to my most recent sort of research interest, and it's perhaps a more interesting application of the method in that it allows a tree cavity to be scanned and then 3D printed in multiple longitudinal cross sections, which essentially act as frames in a conventional beehive. And you can see here that I put a bit of wax foundation at the top there. And the idea is that you can inspect these frames as you would a normal colony and see how different cavity geometries influence behaviours like home construction and nest organisation and ultimately colony survival. So this is what I'm going to be testing out in my free time this summer. Now I'm just going to give a quick overview of the presentation. So in the first part it showed that only a small percentage of honeybee colonies in Europe are free living as a whole, but there are hotspots, for example in southeast England, southern England, where free living colonies contribute significantly to local gene pools. And they're not just swarms that have escaped from apiaries, and they potentially represent important genetic material, which we'll talk about more in the second session. As for the second part of the talk, shown that it's possible to quantify the quality and quantity of available nest sites. And I've shown that in most places the density of suitable cavities probably exceeds that needed to house a free living honeybee population, and that other factors like food availability are likely more important. But this is not conclusive. The question is still open, and I'd love to discuss more about that in the second session or after this talk. So with that, I'd just like to thank firstly my PhD supervisor, Francis Ratnieks, and the rest of the team at the University of Sussex. And I'd also like to thank the CB Dennis British Beekeepers Research Trust for funding my PhD. And thank you for listening.
How beekeepers can support wild living colonies, what would you say?
Well, I'd say by using locally adapted bees and trying to maybe introduce more natural selection into the apiary rather than artificial selection for some of the traits that beekeepers like, selecting for survival more than anything. Will also increase the likelihood that colonies can then survive in the wild. So I think that's partly the issue in Germany. The reason that the free living colonies have such low survival is because the bee that's favoured by beekeepers just isn't able to survive in these places. Like in some parts of Germany, the native bee, the dark bee, has been completely replaced by Carnolian bees. And studies have shown that these bees from southern Europe are really not suited to survival in northern latitudes. They'll choose cavities which are just way too small to store enough honey to survive winter. And they're also go through the honey stores a lot faster than the dark honeybee, which is more conservative. So I think just using a bee that's suited to the environment, it seems simple, but I think that's probably the most effective way that beekeepers themselves can help free living colonies.
Thanks.
It looks like a really fun PhD, so amazing. Just got a few questions. One is, you could just remind us of the veteran trees that you surveyed in the southeast, but what percentage did contain suitable potential, kind of sites or whatever? Do you have any data for the southwest area? And as from somebody recently who's thinking of doing a project to veteranize trees, I saw something. I don't know that process at all. But do you have any sort of suggestions regarding, how they might go about that in order to provide, suitable colonies in the future for living bees?
Yeah, okay. So for the trees I surveyed, we estimated that around half of them had suitable cavities. But this is almost certainly an overestimate because I didn't have this 3D scanner back then. So we were just basically judging the suitability from the size of the entrance, which you can see from the outside. But I do think that, the trees listed on this database, a lot of them really do have quite suitable cavities, but that's, yet to be quantified with any certainty yet. Trying to remember what your other question. So we've only just started the project in the southwest, but one of the places that I've been looking for free living colonies are these sort of landed estates, and as I've shown on that map, there are still quite a few in the southwest. I mean, not as many as in the home counties, and places like Killerton House, these places that are open to the public, high density of veteran trees, are great places to look for free living colonies. And we're basically, instead of me doing all of the surveying, we're trying to get beekeepers and the general public involved in the bee research, because it's it's a lot of fun, and I think people will enjoy it. And yeah, it's interesting that you mentioned veteranization, because that's actually an area of research that I got into but didn't mention here because it wasn't completely relevant, and it was also based in Spain. And we were looking at sort of unintentional veteranization, from where the people that harvest cork off these cork oak trees were accidentally leaving wounds in the trees during the harvesting process, and then these wounds would then turn into cavities over time, which could then be occupied by cavity nesting species, including honeybees. So some of the best ways to veteranize a tree are just through routine maintenance, like pollarding as well. A lot of studies have shown that pollarding creates habitats for cavity nesting species. Or you can cut out just go straight to creating a cavity in a tree without relying on random processes like decay, which is like a traditional way of keeping bees in, like the Russian bee forests. Yeah.
I think I'm doing my work on National Trust Booty.
Really? Okay. Yeah.
I don't know if you've been in touch with them regarding surveying their biology.
No, it's not a place that came up. I have to add that to the list.
A question's come in the Zoom. You may have already said this, but what percentage of managed colony swarms survive in the wild?
So the thing is that we don't know where the swarms have come from. So they could have just as likely come from free living colonies, especially on these estates where there's relatively high density. But yes, I guess that's a good question, because there is a difference in survival between established colonies that have already survived a winter compared to founder colonies, which are in their first year. And Tom Seeley showed that there's a huge difference between survivorship between established and founder colonies. He showed that established colonies will survive 80% of the time, whereas the new swarms only 20% or so. But from my research, I showed that the difference isn't that significant. So there is a difference, but it wasn't actually statistically significant. And this could be due to a sampling bias, but it could also be due to the fact that the winters in southern England just aren't as severe as they are in upstate New York, where Tom Seeley was doing his work. So yeah, they're less likely to survive, but you know, it's not as bad as they've got it in places with really long winters.